filtration, and specialized transport. Filtration is a passive process where small water-soluble drugs pass through tiny pores in cell membrane or between cells. It is enhanced by fluid movement driven by hydrostatic or osmotic pressure, which is common in capillaries, especially in kidney glomeruli. Only lipid insoluble drugs small enough to fit through these pores can cross by this mechanism. Specialized transport. All cell membranes contain numerous carrier proteins which are transmembrane transporters that facilitate the movement of vital ions, nutrients, metabolites, neurotransmitters, and other molecules across the membrane. Unlike ion channels, which form open pores and allow passive flow of ions, carrier operates via a two-step mechanism. First, they bind a specific substrate, either an ion or an organic molecule, on one side of the membrane. Second, through an alternating axis conformational change, the carrier shuttle translocates the bound substrate to the opposite side, releases it, and then returns to its original shape. Depending upon the requirement of energy, carrier transport is of two types. First, facilitated diffusion uses solute carrier transporters, a family of transmembrane proteins, to passively move substrates along their concentration gradient from areas of higher concentration to lower concentration without the need for energy input. Second, active transport requires energy and moves solutes against their electrochemical gradient from areas of lower concentration to higher, resulting in selective accumulation on one side of the membrane. Active transport can be primary or secondary depending upon the source of the driving force. Primary active transport uses direct energy, typically from ATP hydrolysis. Transporters, often called pumps, bind the solute on one side of the membrane, hydrolyze ATP to fuel a conformational change, then expel the molecules to the other side. Common examples include the sodium-potassium ATPase, which move 3 sodium ions out and 2 potassium in per ATP. Secondary active transport does not use ATP directly. A transporter binds both the driving ion and the substrate. As the ion flows down its gradient, the substrate is carried against its gradient. The sodium-glucose cotransporter in the gut is a secondary active transport mechanism. It uses the energy stored in the sodium gradient created by the sodium-potassium ATPase to move the glucose into intestinal cells against its concentration gradient without directly using ATP. In symporters, both molecules move in the same direction. Example, sodium glucose cotransporter in the gut, while in antiporters they move in opposite directions. Example, sodium calcium exchanger. For more such videos, don't forget to like, share, comment and subscribe.